This is an area that he has stayed active in, and is currently with EnzyPep, and we're giving a presentation on enzymatic ligation technologies as adjuncts for the assembly of peptides and proteins.
Thank you. Thank you for this kind introduction, and also big thanks to the organizers for this opportunity to come all the way from Europe here to give this presentation and present our technology, which is already going on for 5 years, but we kept it secret for a long time, and now we're really trying to push it forward and get people to use our enzymes, which can be used for ligation of peptides and the synthesis of conjugates. I forgot my business cards this week, so anybody who I disappointed, my email address is here. And just to be sure, I also put it on the last slide, so then if you want to contact me, it's just timo@enzypep.com. So one slide about EnzyPep and where it's located. It's in Europe, in the Netherlands, and we are at the total south bottom of the Netherlands. We started 5 years ago as a spin-off company from a larger company called DSM, private investors and 2 investment funds, and the core team is me, more for the chemistry. Anna, she's responsible for the proteins and the mutagenesis, biochemistry. Marcel Smit is mainly working on cyclic peptides. So we try to develop commercial process, also scalable processes for the manufacturing of pharmaceutical peptides and conjugates. So I tried to summarize our technology in one slide, so I don't want to give a long introduction about our enzymes and how we discovered them. But what we actually do, you take a peptide or protein A and a peptide or protein B, and you couple them together. That's it. And we produce the enzymes which can do this. So it's an enzyme that couples the C terminus of a peptide to the N terminus of a peptide. And we use a normal ester, and this ester is cleaved off as an alcohol, and you get a natural peptide bond. So it's almost the same as native chemical ligation, because the enzyme makes a thioester with your peptide, and a thioester is prone for nucleophilic attack by an amine. Only you don't need a cysteine. So it's native chemical ligation at any amino acid. So on this side you can have any amino acid, and on the N terminus also any amino acid. So in theory you can make every possible peptide, and it's traceless. So you don't see an enzymatic footprint, it's just the natural peptide bond as a product. It's also much faster than native chemical ligation, and it's not an equilibrium like many enzymatic reactions. So you don't go back, you only go towards the product, and that's it. So in theory it's a 100% yielding ligation reaction. And if you make a long peptide via solid phase chemistry or modified protein or a conjugate, it's usually always better to do a fragment-based approach than a full solid phase approach, as long as you have a very efficient ligation strategy. So our enzymes usually ligate to peptides in over 90% efficiency, leading to a higher overall yield for your process and a better purity of the product. So we split our company a bit in two. One of it is mainly focusing just on the synthesis of linear pharmaceutical peptides, but we also use our enzymes for conjugation to proteins. So that's more the second part of the presentation, and also antibody drug conjugates and those kind of things. So 5 years ago, when EnzyPep started, we discovered our first ligase, which we called peptiligase. And essentially it's a combination of 2 enzymes from literature, one very stable and robust enzyme scaffold, which can handle organic solvents, and the enzyme still does its work. It can handle disrupting agents or reducing agents, and it still remains active. And we added some mutations to make this a ligase instead of a hydrolase. So you take a very robust enzyme and you make it into a ligase, and that's what we also published. So it catalyzes this peptide ligation in water, at the almost neutral pH, and you use fully unprotected peptide fragments. So lysine side chains do not react, or aspartic acid or glutamic acid do not react. It's only coupling the C terminus of a peptide to the N terminus of another peptide. The reactions are quite fast, usually 30 minutes or 1 hour, depending on how much enzyme you add. You can use higher temperatures, and at the coupling position there's no racemization at all. We usually use these type of chem esters, we call them. They mimic a peptide bond and they are well recognized by the enzyme, but you can also use thioesters or HMBA esters or other esters as starting material. And these type of esters, you can easily make them via solid phase synthesis, and it's also something we published. So you use just the standard solid phase synthesis to make these peptide ester building blocks. So starting from peptiligase 5 years ago, we thought we want to create one enzyme that can couple every peptide you desire. So peptiligase, here is the ligation site. It recognizes 4 amino acids of the ester peptide, and it recognizes 2 amino acids on the nucleophile side. So in total, the enzyme recognizes 6 amino acids. Whatever comes after doesn't really matter. And we try to broaden these pockets to make a totally unselective enzyme. So by mutagenesis, we engineer decreasing selectivity towards an enzyme which we call Omniligase, which can couple virtually anything. But during our search to Omniligase, we also found a lot of selective ligase, which can only couple negatively charged substrates or only large hydrophobic substrates or small ones, and we call this selective ligases. And these selective ligases can be designed towards the sequence you want. So just if you have a particular coupling, these selective ligases can be optimized for that specific coupling, and I will tell some advantages later on. So now to the applications of the Omniligase and the selective ligases. First, the application to peptides. So if you take Omniligase, which can couple anything, and you want to make this AB peptide, you have one problem, because you have an amine here and you have an amine on the fragment A. So you have to use protecting groups, because otherwise what could also happen is that you have polymerization reactions and the wrong amine is coupled, so you have to use a protecting group. However, this is not what we observed, this polymerization. But what we observed was that very efficiently by Omniligase, peptide A is usually cyclized. So cyclization is often preferred over coupling or polymerization. And we decided to investigate the cyclization of peptides further, because cyclic peptides are interesting molecules on itself. So Omniligase can be used to almost quantitatively cyclize linear peptide esters. And we first took a look at how long the peptide should be, and we found that you need to have more than 12 amino acids for a very efficient cyclization reaction. But as soon as you go over these 12 amino acids, cyclization is almost quantitative. And when we go longer and longer and longer, and we have seen now up to even 150 amino acids long peptides can be cyclized by Omniligase. So we don't know if there's any length limit or not. Here's just an example. It has no added value, but to show that we don't need an enzymatic recognition sequence or we don't need anything special, we decided to cyclize exenatide. And of course you don't have to cyclize at the 1 or 39 position, but you can also cyclize on another position or there. And we chose to cyclize on the 21 position, but still the product is of course cyclic exenatide. And here you see that the linear precursor is very efficiently cyclized to this 39 amino acid long cyclic exenatide. And what we could even do was take the Omniligase, put it in a reaction vessel, and then dose the linear peptide to the enzyme, and it continuously cyclizing the product, so you can go to very high product concentrations up to 100 milligram per milliliter of cyclic peptide product. We have also shown that if you have a good recognition sequence for enzymes on the C-terminus and the N-terminus, it doesn't really matter what you put in between. So this can also be lysine linked with a side chain, or even PEC chains can be added here, or DL amino acids, and still the cyclization is very efficient. A few more complex peptides we cyclize, and also we with other ligation technologies. So for instance, bicycle therapeutics use this scaffold to make bicyclic peptide. And we've shown that with our enzymes we can first do this head-to-tail cyclization, then add this tribromine and make tricyclic peptides in a very efficient way. And also this symposium we've seen a few times the cyclotides and the cysteine-rich peptides coming by. So that was also very interesting for us to try our enzymes here and to see if we could really come up with a scalable process for the synthesis of cyclotides, because usually now native chemical ligation is used for the cyclization, or the peptides are produced in plants, which is difficult to scale up. So we also tried to make these cyclotides using our enzymes, and that actually worked very efficiently. So once again, you make the linear precursor with the chem ester with standard solid phase chemistry, and on gram scale we could cyclize this using our enzyme and then do the natural folding towards the product. And we, together with the University of Maastricht, we did some NMR studies to show that we really have the correct folding and the correct conformation of this cyclotide MCOT12, and it's possible to immediately purify this by PrEP HPLC, and you have a high overall yield. What we also did was immobilize Omniligase and put it in a flow reactor, and then pump the linear peptide over this flow reactor, and then cyclic peptide product just comes out. So it's possible to use these enzymes in a sort of plug flow reactor, and only your product comes out of the bottom. So that's a summary about the cyclization using Omniligase, and this Omniligase is also commercially available. So if you want to use this enzyme for peptide ligation or for cyclization, it's, we sell it as an Omni kit. Also you have then a peptide ester and a peptide amine, so you can do a test coupling reaction, see the efficiency of the enzyme. In Europe you can buy it via Iris Biotech, and in the United States via Peptide Solutions, and soon also via Sigma Aldrich. But besides, if you take now Omniligase, as I mentioned earlier, you would need an N-terminal protecting group if you are really going to make longer peptides, because otherwise you get cyclization or polymerization. We often use the phenylacetal protecting group, which can be removed with another acylase. So it's also an enzymatic deprotection, and then you could couple your next protected fragments using Omniligase, and again do, so it's a more protection deprotection scheme. But much easier would be if you take a selective enzyme which only recognizes this amine and does not recognize the other amine. So selective enzymes can be used for peptide ligation without any need for protecting groups or deprotection steps. So no protection, just free peptides and one desired product. And then with a second selective ligase, which can recognize this amine but not the other one, you can do your next coupling. And I will give you some examples of these type of ligases. They are not commercially available because we don't have a ligase for any sequence, but you can always contact us if you need a specific ligase for a specific sequence. One example is thymosin alpha-1. It's a 28 amino acid long peptide. It's quite challenging to make it via SPPS because there are so many charged residues with bulky protecting groups. And we decided to make this 28 amino acid peptide from a 14+14 fragment condensation. And 5 years ago, when we used peptiligase for this reaction, the ligation yield was a bit disappointing. And we saw that it probably came because you have a positive charge here on the ester fragment and a negative charge there. And these opposite charges, they didn't want to couple together. So we decided to improve our enzymes specifically to recognize these charge residues by mutagenesis in the pockets, in the recognition pockets, and putting opposite charges in the pockets of the enzyme to improve the coupling efficiency and yield. So over 5 generations of enzyme engineering, we could see that the reaction speed was improved a 5-fold, but more importantly, the reaction efficiency, so the coupling efficiency, from a few percent of product went to more than 90% of product. So we can successfully design our enzymes towards a specific product and a specific coupling reaction and improve them for a specific coupling. And here, this is then what the end product looks like. So we did this on gram scale using crude fragments. So this is eventually what your crude product looks like. You have a small excess of the amine fragment and a little bit of CAM ester hydrolysis. And you see that many of the impurities are also very well separated from the ligation product peak. Nice concentration of thymosin product. And if you look at the ester and the conversion to the product, it's then more than 90% efficiency. So we could use this crude reaction mixture directly for preparative HPLC purification to yield the product in a good overall yield, which is almost twice, twofold better than if you do the full solid phase synthesis. A similar example is exenatide, a bit longer peptide of 29 amino acids. We tried to make it using a 21 plus 18-mer fragment condensation. And then once again, the enzyme has to couple the ester part to this amine and isoleucine, and it does not couple to this histidine glycine. So you need a selective enzyme which couples to this N-terminus but does not couple to the other N-terminus. And also this enzyme was quite easy to find and produce. And here you see the reaction mixture. So the ester fragment, it's totally consumed by the enzyme, going to 0. And the amine fragment, we use it in a bit of excess, so there's a little bit of left. And in 1.5 hours, you have almost full conversion to the exenatide product. We scaled this fragment synthesis now with partners to multi-hundred of gram scale. The enzymatic coupling has also been done on multi-10 gram scale, as well as the purification. And we did not find any new impurities related to this enzymatic ligation technology. And the product proved also to be within the specification of the producer. So these enzymes can be used to efficiently synthesize pharmaceutical peptides at scale. In a collaboration with CEM, we also showed that you can have access to long and complex peptides with incredible speed. So they made some fragments for us on the microwave synthesizer in a matter of hours, and our enzymatic ligation in a matter of 2 hours. So within 1 day, you can synthesize exenatide and purify it by preparative HPLC to have your product. So fast synths of the fragments, and we use them just crude, non-lyophilized, just the precipitated peptides. You ligate them together to your product and then do the final purification. And now we are moving this on to more complex peptides or proteins, so you can have access to proteins or modified proteins within a short amount of time. Thymosin and exenatide are just 2 examples of the selective enzymes, but what we now developed is a computer model where all the data of our variants is in. So if you have a specific sequence you want to couple, we can put this sequence in the computer model and it will predict the optimal selective enzyme, and we can produce and purify this enzyme within a matter of weeks. So now, usually, if someone requests a selective enzyme, we can produce it in a matter of weeks for the coupling, for the sequence that you desire. And with these selective enzymes, it has even proven possible that we do 3 fragments in one pot reaction with 2 selective enzymes. So one enzyme that only does this coupling and the other enzyme only does the second coupling. So you get only one product in one pot reaction. So now from the peptides we move more to the proteins and the conjugates. Because many people ask us, we only focus on the synthesis of pharmaceutical peptides. How long can you go? What's the limit of this ligation technology? And actually we didn't know. So what we first tried was we took the X10 polymer, which is also used for half-life extension of pharmaceuticals, similar to the pacylation story we heard yesterday. Extremely long peptide, almost 900 amino acids long, and we've shown that if we take a model peptide, we are able to couple this C terminus to the N terminus of a big X10 polymer, making products of almost 900 amino acids long and still in good efficiency. So this demonstrates that there's almost no length limitation to this ligation technology. This is the longest peptide we tried so far, but we think we can even go longer. But of course proteins is a totally different story because they have they are folded and they have a nice tertiary structure. If you look at the protein, you could couple to the N terminus of a protein, or you could couple to the C terminus of a protein. But for us, coupling to the N terminus is much easier because for the C terminus we need an ester of a protein. So first studies we did was coupling peptides to the N termini of proteins. And this is one example where we took human serum albumin. It's more than 500 amino acid long protein. And we took exenatide CAM ester just as an example, so a very long peptide ester. And with the Omniligase we could very efficiently do this coupling reaction. And we showed that if you use only 4 equivalents of this exenatide CAM ester, the coupling to human serum albumin is almost quantitative. So you don't need a huge amount of your peptide, but 4 to 5 equivalents can lead to quantitative coupling to a protein. And we also shown that it's compatible with other ligation technologies. So we could also take a peptide with a click handle, couple that to human serum albumin, and then do a click reaction, for instance, with a fluorescent molecule to get more ligation products. And of course, you can also use this to ligate or click proteins with their N terminus to each other. And here, with the human serum albumin coupling, we use a recognition sequence for our enzymes, but we have also shown that in most cases, this is not needed. So you can just take a natural protein without an N-terminal extension and then do your enzymatic coupling to it. Another thing we developed is the double-headed CAM ester. So it's a peptide and a linker which has 2 esters, one on the right side and one on the left side. And with our selective enzymes, we can first couple one ester to the N-terminus of a protein, and then with a second ligase, we can take the second ester to another protein. So you can link proteins to proteins using this double-headed CAM ester. One of the holy grails is coupling reactions to antibodies. So that's also something we are investigating. And first, we took only a Fab fragment with 2 extensions on the N-terminus to make the coupling reaction with our enzymes easy. And we chose the amino acid as such that we can, with our enzymes, selectively couple to the heavy chain a molecule, or selectively to the light chain a molecule, or you can couple to both chains at the same time if you take the Omniligase. And here again we took the exenatide example and selectively coupled it to the light chain of a Fab fragment. Then we went one step further and we just took a full unmodified antibody and coupled a fluorescent peptide to it. And even here we show that with selective enzymes we can selectively couple, for instance, to the light chain of an antibody, and with other enzymes we can couple to the heavy chain of an antibody. So depending on the sequence of your antibody, we can do selective couplings to the N-termini of unmodified monoclonal antibodies. As a final example, similar to the heavy chain and the light chain, you also have dimeric peptides like insulin, which have an A chain and a B chain, and with selective enzymes you can couple, for instance, only to the A chain, and with another ligase you could couple a peptide to the B chain. So with these selective enzymes you can discriminate between different N-termini and couple your peptide or protein of interest to the chain you desire. Or if you take the Omniligase, which is very broad, you could couple the same peptide to both chains at the same time. The C-terminus, it's something we are also looking at, but as I said before, it's more difficult for us because you need to produce a protein with an ester on the C-terminus. But there are some technologies known to produce proteins with a C-terminal ester. For instance, by intein expression. Via intein expression, you can make a protein with a C-terminal thioester, and you can, you can use this thioester directly with our enzyme. So immediately take this, we did this with maltose binding protein. You produce it as an intein, make the thioester, and then immediately do a ligation reaction. And what we also did, ligate a small peptide with a cysteine and a chem ester, so native chemical ligation to make a protein chem ester, and then use this chem ester to couple another peptide fragment. So via intein expression, we can also couple peptides or whatever you like to the C-terminus of a protein. So to conclude, these peptidigases, they are usually very fast and they are powerful enzymes for peptide ligation. They couple the C-terminus of a peptide to the N-terminus of a peptide, and you don't see an enzymatic footprint, you just get your natural product. It's irreversible, so ligation yield is often very high, and you don't have to stop the reaction at exactly the same, at the perfect time, because it's not going down again, it's just going to product and that's where it stays. You do need a C-terminal ester, which can be a chem ester, but also an HMBA ester or a thioester, or whatever ester you like. We often use chem esters because they are quite easy to make via solid phase synthesis, and they are well recognized by the enzyme. The reactions are fast, and also the concentrations can be high or low if you make conjugates, but they still work. So you can use it to synthesize pharmaceutical peptides or to make cyclic peptides at scale, and also for novel biologics or conjugates, these enzymes can be used. So I would like to thank a lot of people, but I did not put all the names on the slides. So of course the EnzyPep R&D team, especially Marcel, who did all the cyclization reactions and the work on thymosin, and Anna Toplak, who is responsible for the mutagenesis and the protein production and purification. I think every new technology needs a few real believers in the field, and sure, Rodney Lex is a believer of this technology, so I would like to thank him for really trying to push this forward and get people to use our enzymes and incorporate this technology into their companies, and Peter Kwaadvlieg from DSM and our investors. So I would like to say we have one open R&D position, so if anybody is interested, more than welcome. We also take students, and if you have interest in our enzymes, the Omniligase or selective enzymes, feel free to contact me. So once again, my email address, and thank you all for your attention.
Well, thank you for a terrific talk. I'd love to apply for that position. It sounds fascinating.